Hello everyone, it's me Aisha. I'm going to review this paper which is Chromosome-scale haplotype resolved assembly of human genome. Here is the abstract: to study complete genomes along their complex genetic variations. There is a need to generate chromosome-scale haplotype-resolved assembly. So here authors present a novel method which is known as DIP-ASM. It is a diploid assembly method which were applied to four public human genomes that I have shown in red color. This method have several features: that it can complete genome assembly within 1 day per individual, and the NG50 value is up to 25 MB, and it can phase 99.5% of heterozygous sites at 98 to 99% accuracy rate. Question: that's why we need chromosome scale phased or haplotype resolved assembly. The answer is that it is very important for the discovery of structural variants and also for the identification of new transposons insertions. It is also very important for the discovery of highly polymorphic regions that are medically important, such as HLA and KIR regions, and it is also very important for the study of allele specific DNA methylation and gene expression. Several traditional de novo assembly algorithms that reconstruct genome assembly as a haploid genome, but for a diploid genome such as for human, this result in the form of loss of heterozygous variations in the genome, and also the assembly errors in regions diverge between haplotypes lead to inflated assembly for species with high heterozygosity. So many methods has been proposed to do phase assembly, such as Falcon unzip, supernova and Falcon phase. These are some methods with their limitation. The first one are the Falcon unzip and supernova, which are unable to phase through centromere and long reads. The second method is the Falcon phase method, which use high C data, and it is unable to achieve chromosome long phasing with Falcon phase method. The third method is the trio binding method, which is unable to resolve regions heterozygous in trio. So there is a need to generate phased assembly without any prior knowledge of parental samples. The latest method, which combine high-Phi and high-C data, it can construct two haplotypes in a diploid individual without any pedigree information, and it has ability to accurately resolve highly polymorphic regions of biological importance. So here are some limitations: it cannot work with noisy long reads at present, and it starts with unphased assembly that may miss highly heterozygous regions involving long SVs. So here is the outline of the phase assembly algorithm, which uses high C and high Phi data for unphased contigs, and after making unphased scaffold it can turn into the phased contigs with the help of several bioinformatics tools. Here shows the application of phased assembly. The first one shows the local sequence divergence in comparison to the reference HLA haplotypes at the top and to the KIR haplotypes at the bottom region in GRCH38 reference genome. So the second part of this figure shows the structural variant density on chromosome 1 for all the four genomes that have been studied in this research. Some future perspective of this method, that this method will facilitate high quality precision medicine and studies of individual haplotype variation and population diversity. Its ability to generate phased assemblies without using a reference sequence will enable the unbiased characterization of human genome diversity, and surely it will construct a comprehensive human pangenome, which is the current goal of Human Genome Reference Project. There are some suggestions from my side. We can upgrade PGP1 assembly by using deep ASM and other latest method, and we can also compare the old version of this assembly with the new version to know the characteristic differences. For this we can try to phase long reads and to resolve long haplotypes and to reduce the gaps. for making a better version of PGP1 assembly.